So I think we'll go ahead and get started. I just wanted to make a few remarks here so that we can introduce our speaker and give our guest here the full amount of time to present his research. My name is Kevin Lang. I help organize the Research, Innovation, Discovery, and Education seminar series for the College of Vet Med. One of the things we wanted to do with this seminar series is provide a space that we can highlight the research of collaborators from the faculty within our college. And so this opportunity presented itself to have Dr. Chae present his research during his short visit here to the University of Minnesota. And I just wanted to first thank the research office, Alonso and Kinsey and Jessica and everybody else in the office for helping coordinate this talk and also this seminar series. And I wanted to welcome all of you. Thank you for being here. Please look forward to the future of this seminar series. So next week, February 12th on Wednesday at noon, we're going to be having two internal speakers from within the college present their research. In March, we're having a panel discussion on navigating collaborations and within academia and externally. So we have a pretty good lineup for this semester, so please look for the second Wednesday of every month for the RIDE seminars. So I'm going to keep it quick so that we can give our speaker the full time. So I will have Dr. Peter Larson come and introduce our speaker.
Thank you so much, Kevin. So again, I'd like to welcome everyone to this first joint CVM RIDE and International Institute of Biosensing special guest lecture. I'm Dr. Peter Larson, as Kevin said, Associate Professor in the Department of Veterinary and Biomedical Sciences. I'm also on the Scientific Advisory Board for the IAB. So I'd like to begin by thanking Tanya Seiler on our team for helping with the logistics, also Kinsey from the CVM Research Office, and also Dr. Patricia Kashiar from who is Director of the IAB. So the IAB's mission is to strengthen international biosensor research in order to help address critical issues facing human, animal, and environmental health. This mission led to an exciting opportunity last November when I visited Seoul National University in Korea with Professor Sang-Hyun Oh, a faculty member of the College of Science and Engineering and founding member of the IAB. There's a rich tradition of collaboration between Seoul National University and the University of Minnesota dating back to 1955. It's really fantastic. Our goal was to expand on that tradition by uniting our colleges of veterinary medicine and to identify opportunities for collaborative biosensor research. As a direct outcome of our visit, Seoul National University awarded Professor Chae's team a grant to travel here and to initiate joint research on tick-borne pathogen surveillance using nanopore sequencing technology. This research strongly ties together Seoul National University with our IIB, our CVM, and also the School of Public Health through Dr. John Oliver, an expert in global tick-borne pathogens. Also, a special thanks to Dr. Evan Kipp and Lexi Frank for leading our nanopore sequencing workshop with Professor Chae's students. We are collectively honored to host Professor Chae and his students, and we look forward to continued collaboration. Professor Chae is a world-renowned expert in vector-borne diseases. He has published over 250 peer-reviewed manuscripts. I think one today also, just today. It's amazing. He's significantly advanced tick-borne pathogen research across Korea and Asia. Please join me in welcoming Professor Chae, who will share his latest findings on SFTSV in Korea.
Howdy, everyone. Professor Peter Larson, thank you for introducing me and arranging this special lecture at University of Minnesota today. I'm a veterinarian, but I teach veterinary internal medicine, especially large animal internal medicine. But my research focuses on the control of ticks and tick-borne disease. Today, I'd like to talk about the tick-borne severe fever with the Thrombocytopenia Syndrome Virus. It's a very long name. SFTS virus. My presentation will include an introduction, a brief description of the SFTS virus in ticks and animals in South Korea. Transmission, especially for the secondary transmission, secondary infection, and conclusions. Ticks are one of the longest surviving animal species on Earth, and are vectors for transmitting various pathogens to humans and animals. Recently, ticks have been known to be vectors of the virus that cause SFTS, a disease with a very high mortality rate in Asian countries. Due to global warming caused by climate change, the tick habitat in Korea may expand, and ticks are expected to continue to pose a threat to human and animals in future. Two studies have reported that the distribution of ticks and the spread of related disease due to global warming and increasing. In this slide, you can see the spread of ticks and tick-borne Lyme disease in North America and Northern Europe. The authors believe that this cause is related to climate change. Another paper reviewed the investigation of tick-borne disease for the past 10 years in Slovakia, one of the European countries. The authors explained that the increasing trend of these tick-borne diseases is related to global changes, as well as advanced in disease diagnosis technology. As you can see in these researchers, spread of tick-borne disease is increased by climate change. This slide shows have climate change spread zoonotic disease like tick-borne disease. This is an infographic of the dangerous cycle that climate change creates for zoonotic disease. Humans expand the wildlife-urban interface, and climate change increase homogenized habitats, creating areas of increased biodiversity where novel species interactions can occur. These interactions present an increased risk for the spread of zoonotic disease. Environmental changes due to climate change are increasing the density of wild animals and ticks in South Korea. National Institute of Meteorological Research in Korea reported that Korea climate is expected to change to a subtropical zone until 2100, excluding high mountain areas. Currently, due to nature conservation and animal protection activities in South Korea, the density of wildlife, wild animals, and tick vector has increased. In addition, as people's outdoor activity increase, the chances of being exposed to tick have increased. Therefore, South Korea is expected to be a country where tick-borne infectious disease can increase from an environmental perspective. There are many factors of disease transmission, especially life cycle, food chain, vectors, reservoirs, and migratory birds. Migratory birds bring along ticks and pathogens and pass over the borders to other countries. Recently, it has been reported that transmission of the SFTS virus may be related to migratory birds flyway in Asia. Therefore, we need more detailed studies on the relationship between migratory birds, ticks, and SFTS virus. This is a map of the routes for migratory bird. Most migratory birds move from south to north, from north to south, depending on the season. Korea is an important stopover land for migratory birds. Korea is located in the middle of the East Asia Australian flyway, which is a stopover site for migratory birds. Research is going on in South Korea to investigate the spread of the SFTS virus by ticks on migratory birds. Bird Research Center in the Korea National Park Research Institute collected ticks when catching birds for the banding program. We discovered several new species of ticks collected from migratory birds that have not been reported in South Korea. And several pathogens, Anaplasma, Borrelia, Bartonella, Rickettsia species were also detected. Therefore, we will be able to establish preventive measures based on the study on the interaction between migratory birds, ticks, and SFTS virus in the East Asian flyway region. The Asian long-horned tick, Haemophilus longicornis, is the competent vector for the SFTS virus. Haemophilus longicornis originated mainly in Eastern Asia and invaded many areas like Australia, New Zealand, and the Pacific Island, and was recently introduced the eastern part of the United States of America. This species is characterized by high adaptability to a wide range of temperature and can reproduce parthenogenically under stressful conditions. Migratory birds are important hosts of Haemophilus longicornis and are thought to be responsible for its unexpected invasion and introduction into new area worldwide. Now, I will talk about the SFTS virus. SFTS virus is a group 5 negative single-strand RNA virus. This virus species was finally released, Panda virus daviensis, the species name, by the International Committee on Taxonomy of Viruses in 2023. The virus was 3 segments, L, M, and S, and the genome size is 11,490 base pair, very small size virus. The SFTS virus is a newly recognized hemorrhagic fever disease found throughout East Asia, China, Japan, and Korea. The SFTS is an emerging infectious disease first developed in China in 2009. In 2011, the first human infection with the SFTS virus was officially reported in Western China and then reported in Japan and Korea in 2013. Recently, the SFTS virus detection area has been continuously expanded Asian countries. This slide is about the SFTS virus detected in ticks collected from migratory birds. And it is the first paper published by my research team in 2024. We collected Haemophilus consina and Ixodes turdus ticks from the Emberiza spodocephala, captured in Daecheongdo Island, and Oliva-backed pipit captured in Heoksando Island in Korea, respectively. The SFTS virus genotype B2 was detected in all of these ticks. 2 Tracking of these captured birds and ticks, the birds were identified as summer migratory birds from southern China that entered Korea in March and April. Haemophilus consina and Ixodes are tick species that inhabit China and Southeast Asia. We had suspected that the SFTS virus could have landed in South Korea through the tick carried by migratory bird from Southeast Asia to South Korea. But our research team has discovered it for the first time. The SFTS virus transmission to humans commonly occurs from virus carrying tick bite. The SFTS virus then invade the lymph node, targeting immune cells such as B cells, impairing the host immune response from invading the pathogen. The SFTS virus causes systemic circulation, viremia, cytokine storm, and severe inflammatory response syndrome after the further replication. Thrombocytopenia is a hallmark of the SFTS virus infection. Disseminated intravascular coagulation and endothelial damage caused by cytokine storm lead to multi-organ dysfunction, reflected by the elevation of liver, kidney, and heart serum markers. SFTS virus infected person have a severe fever, leukopenia, thrombocytopenia, and elevated liver function values. If doctors did not earlier diagnosis, a patient may have multifunction organ failure and die. Currently, the genotypes of the SFTS virus are classified into 6 genogroups, from genotypes A to F, and the genotype P is divided into subgenotypes B1, B2, and B3. In South Korea, genotype P is the most common, according for about 77% of humans and 68% of animals. All genotypes of human SFTS virus have been found in South Korea, but only genotypes B, T, and F have been found in animals so far. The genotypes A and F of the SFTS virus showed low virulence in the mouse infection studies. Therefore, we need a specific study on the virulence according to the genotype of the SFTS virus. This slide is the geographic distribution of the SFTS virus. The genotypes of the SFTS virus in China and Japan and different from each other. And both the Chinese and Japanese genotypes are found in Korea, the middle of the site in Korea, between the China and Japan. In 2020, my lab first discovered the new subgenotype P4 of the SFTS virus in cats and cows. These findings suggest that this SFTS virus has a genotype that has not yet been transmitted from ticks to human or animals to humans. Or it is thought that we need to do more research to find out whether a genetic mutation of the SFTS virus in host animals in the course. In 2021, one of the SFTS human patient in Guni-gun in Gyeongsang-buk-do, South Korea. Therefore, the Korea Disease Control and Prevention Agency tried to multi-ministerial joint surveillance with National Wildlife Disease Control Center. The NWDCC collected blood samples of detached ticks and captured Korean water deer in the same areas as the SFTS patient in Guni-gun. We tried to test for the SFTS virus, and we got PCR positive from Korean water deer, and I'm sorry. Yeah, got it. We tried to test for the SFTS virus, and we got PCR positive from Korean water deer and ticks also. We analyzed the sequence of the SFTS virus. The genotype of the S segment was subgenotype P4. The genotype was identical SFTS virus among human patient, Korean water deer, and ticks. These results demonstrate the life cycle of the SFTS virus in nature, transmitted through ticks and animals, and then transmitted to people in the same area. The ecological habitats of Korean water deer are active while inhabiting within 150 meters of the human living village in rural areas. In this surveillance, we need to better understand the natural ecosystem and approach disease prevention. This case was the result of the cooperation of each department based on the One Health concept. In South Korea, the SFTS virus was first isolated from a human patient in 2013. The number of SFTS infection cases has been steadily increased over 5 years, and then a little bit decrease in South Korea. Since 2013, a total of 1,089 people have been infected with the SFTS virus. And 355 people died until 2023. The case fatality rate was 18.7% in humans. It was higher than any other infectious disease. As you can see from the map of the left, the areas within the highest average annual infection rate per 100,000 people are mountainous areas with high wildlife density. Our laboratory has been conducting research on the SFTS virus in ticks and animals since 2013. Ticks were collected from natural environments and animals, and the SFTS virus was detected in collected ticks and blood samples from companion, farm, and wild animals. Main dominant tick species in South Korea is Haemophilus longicornis, more than 95%, and then Haemophilus flava, Ixodes nipponensis, and Embryoma testinaridium. Ixodes ovata is a very rare species. We collected only one ticks in Deogyu-san National Park in South Korea. We collected also 4 different tick species from wild boars, especially Embryoma testinaridium is a host-specific tick species that resides in wild boars, snakes, and humans. We could not see any Embryoma species on livestock animals. In our study for the detection of SFTS virus antigen from ticks and animal samples, nested PCR and real-time PCR and nucleotide sequence analysis were performed. Also, we have established IFA and ELISA method for antibody investigation against the SFTS virus. We surveyed for SFTS virus in 5 national parks: Mountain Bukhansan, Chiaksan, Udaesan, Deogyu-san, and Wolchul-san. national parks in 2015. We collected ticks on trail and non-trail of the 5 national parks, and then compared the number of collected ticks and the infection rates of the SFTS virus in ticks. There was no statistical significance difference in the number of the collected ticks and detection rate of the SFTS virus infected ticks between trails and non-trail. Wild animals generally do not move on trails during the daytime, but they move or stay along trails at the night time when people are not present. Therefore, ticks are collected on the trails. This table shows the detection result of the SFTS virus. The tick species collected from these 5 national parks, the SFTS virus infection rate of Hampshire longicornis, adult and nymphs was 4.77 overall. Total infection rate in all of the ticks was 3.67%. We detected the SFTS virus from ticks on mountain Gwanaksan and the campus of the Seoul National University in Seoul, South Korea. Mountain Gwanaksan is one of the mountains in Seoul, and the SNU campus is located around Mountain Gwanaksan. Many people enjoy hiking on the mountain. We did not find any ticks from the SNU campus, but I don't know on this campus in the summer season. But we collected 273 ticks from Mountain Gwanaksan and tested them for the SFTS virus infection. We collected 2 different kinds of tick species, Hampshire longicornis and Hampshire flava. The infection rate of adults and nymph tested as a single tick was 3.2%, and the larvae were 2.3%. In the pooled samples as minimum field infection rate. We did study to detect the SFTS virus in ticks collected from Korean water deer and wild boars. The total infection rate was 4.98%. This result was very similar to the detection rate of the SFTS virus in ticks collected from humans. And now I will talk about the detection of the SFTS virus in farm animals, wild animals, and companion animals. First, we collected blood samples from domestic pigs for detection of the SFTS virus in 2013. The pigs were raised in a traditional way near the mountain areas. We collected 240 blood samples of domestic pigs from 8 provinces across the country and detected for SFTS virus infected pigs by nested PCR test. The total detection rate was 1.67%. This table shows infection rates of the SFTS virus from 268 black goats. In 4 different grazing ranches, SFTS virus antigen was detected by nested PCR. In 14 black goats from 3 farms with a detection rate of 5.2%. This slide shows the investigation of SFTS virus from wild boars. The overall average detection rate of the SFTS virus antigen by PCR was 4.3%. And the overall average antibody positivity rate by ELISA was 28.8%. We also detected the antigens and antibodies of the SFTS virus from poultry, chicken, ducks, and wild geese. The wild geese were collected from farm-raised animals. The total detection rate of the SFTS virus from poultry was 2.5%, and the total seropositive rate was 30.3% by ELISA. As a result of sequence analysis, subgenotypes B2 and B3 and genotype T were found from poultry SFTS virus. We investigated SFTS virus infection by capturing stray cats in 7 districts of Seoul. And it was detected in all districts except 1. SFTS virus antigen was detected in the blood of 22 of 126 stray cats by nested PCR. And the overall infection rate was 17.5%. It's pretty high. The detection rate in stray cats at that time was very high percent. We did a study to detect the SFTS virus from Korean military dogs. The training environment of military dogs is highly exposed to arthropod vectors. SFTS virus was detected in the blood of 3 out of 103 military dogs by nested PCR test, showing a positive rate of 2.9%, and the virus was isolated from 1 positive dog serum. We tried to detect the SFTS virus in horses from inland and Jeju Island. The total detection rate of SFTS virus antigen by PCR test in horses from Jeju Island was 2.1%. And the total positive rate of ELISA antigen antibody to the SFTS virus was 24.2% in the inland and 33.7% in Jeju Island. We conducted a study about the clinical and epidemiological characteristics of the patient with SFTS in companion animals. This slide shows one of the clinical cases of the SFTS virus infected dog patient. The patient was a companion dog living in an apartment in Chungju City in South Korea. He was admitted to a local animal hospital with fever and anorexia. Before visiting the animal hospital, he often walked in the grass near Tanggeumdae Park in Chungju City. The scar of the bite was discovered in the lesion around his armpit, left armpit, by the dog groomer. And subsequently, he developed fever and anorexia. Based on this patient's clinical symptoms, blood test, antigen and antibody test, the final SFTS was confirmed, and the virus was also isolated. This study investigates the SFTS patient in dogs and cats from 166 small animal hospital nationwide. In 2018 and 2019, we found 14 dogs with clinical symptoms of SFTS among 448 dogs, and 4 cats with positive SFTS virus among 112 cats. Many SFTS dogs showed clinical symptoms including anorexia, depression, vomiting, and diarrhea. In addition, leukopenia, thrombocytopenia, elevated CRP levels, and liver function tests were observed. The genotypes of SFTS virus infected in dogs were subgenotypes B1, B2, B3, and genotype T. This slide shows confirmed cases of the SFTS in companion cats. SFTS virus antigen was detected in 4 of 22 clinical cases in cats, and 1 cat died. Clinical signs in cat were detected such as anorexia, jaundice, thrombocytopenia, leukopenia, and hyperbilirubinemia. Cat jaundice was observed in most patients with SFTS infection. IFA test also showed strong antibody positivity to SFTS virus. Cat SFTS virus genotypes were identified as subgenotypes B1 and B3 and genotype T and F. We conducted antigen and antibody survey for the SFTS virus in companion, farm, and wild animals from 2018 to 2020. We detected SFTS virus antigen or antibody in a total of 14 animal species. The average detection rate of the SFTS virus in all tested animals was 2.4%, and the antibody positivity rate was 20.5%. Most animal species except cat and goat had high antibody positivity rates. The antibody positivity rate is low because the mortality rate of cats is high in Japan. Goat blood was collected from a slaughterhouse, and the goat had recently been raised in barns in Korea. Therefore, goat raised in barns are less likely to come into contact with ticks. In this map, SFTS virus infected animals were identified all over the South Korea, and Gyeonggi, Gyeongbu, Jeonnam, and Jeju province in particular showed a high antibody positive rate. This study investigated the pathogenicity and potential for intraspecies transmission of experimentally infected dogs with SFTS virus. Secondary transmission by contact has been demonstrated when healthy dogs are co-housed with infected beagle dogs. This case is a direct transmission of the SFTS virus from domestic cats to veterinary personnel. Two veterinary personnel in Japan were infected with the SFTS virus while handling a sick cat. Whole genome sequence of SFTS virus isolated from the personnel and the cat were 100% identical. This result identified nosocomial outbreak of SFTS virus infection in an animal hospital without the tick as a vector. The cat died 3 days after visiting the animal clinic. The veterinarian who treated the cat had a high fever 12 days after first contact and was hospitalized. On day 16 of contact with the cat, the PCR test was positive, and antibodies to the SFTS virus were detected in the blood of both the veterinarian and animal technician on day 28. Therefore, this is a case of the secondary infection of the SFTS virus by veterinarians and animal technicians who had cross contact with cat. This study report is a case of secondary transmission of the SFTS virus from animal to animal and then to human from an undiagnosed febrile dog patient. This paper is a case reported in Japan. It is a secondary infection from a dog patient infected with the SFTS virus to one of the companion dog and 6 people. 4 family members, 1 veterinarian and 1 animal technician. This slide is a timeline of dog to cat nosocomial transmission of SFTS virus in Japan, 2022. The 2 cats that underwent surgery in a room where a sick dog had been euthanized became ill within 9 days of surgeries, ovariohysterectomy and endoscopic surgery. SFTS virus was detected in all 3 animals. Nucleotide sequence identity was 100%. The suspected course was an uncleaned pulse oximeter probe used for the old patient. This report is a secondary human infection from a canine patient infected with the SFTS virus in Japan. A 2-year-old male domestic cat showed lethargy, tonic clonic convulsion, and mucosal jaundice. Upon admission, blood examination indicated severe neutropenia and thrombocytopenia, and ultrasonography revealed diffuse splenomegaly with a honeycomb appearance and abdominal lymph node enlargement, in addition to a decrease in cardiac blood flow indicating a shock condition. Cytology of the spleen showed a cell population composed of immature large lymph node cells with distinct nuclei, suggesting lymphoma. The cat received symptomatic treatments but died 4 hours later. RT-PCR reaction assay of the spleen sample indicated the presence of the SFTS virus. Clinical features of this case that were diagnosed as SFTS were similar to lymphoma. Therefore, guardians of companion animals and veterinary workers should be protected against infection by the SFTS virus. To confirm the infectious virus in the specimens, virus isolation was attempted from serum, saliva, urine, eye, and rectal swab specimens collected during the experimental period. The result showed a high level of viremia in the cats with fetal illness upon SFTS virus infection, with the virus secreted via the blood, saliva, and tears. When antibody tests were conducted on animals infected with the SFTS virus, it was found that IgM increased in the early stage of infection, and was the virus disappear from the body, IgM decreased, and then IgG increased. 4 of the 6 cats infected with the SPL010 strain of the SFTS virus died, all showing similar or more severe symptoms than human SFTS patients, such as fever, leukopenia, thrombocytopenia, weight loss, anorexia, jaundice, and depression. High level of SFTS virus RNA load were detected in the sera. Eye swab, saliva, rectal swab, and urine indicating a risk of direct human infection from SFTS infected animals. An autopsy of the cat that died from SFTS infection in Japan revealed severe jaundice, gastric ulcer, intestinal hemorrhage, and enlarged mesenteric lymph node. This reported study was the clinical and pathological findings of 4 fatal cases of cats with high SFTS viremia level in Japan. These cats showed an acute onset on fever, leukopenia, thrombocytopenia, and increased serum amyloid A and pro-inflammatory cytokine levels. High viral copy numbers were detected in the blood, oral swabs, rectal swabs, conjunctiva swabs, and urine. Histopathologically necrotizing lymphadeninitis, splenitis with lymphoblastoid cell proliferation, and hemophagocytosis were observed in all 4 cats. Immunohistochemistry revealed the presence of SFTS virus antigen on lymphoblastoid B cells. SFTS virus RNA was detected in systemic tissues, including the brain. The present finding provide useful information for understanding the features of fatal SFTS in cats. The authors suggest that to elucidate the mechanisms of severe progress of SFTS cats, as well as its role as a source of human infection, further research is needed. Summarizing Japanese report, a total of 16 people were secondary infected from companion animals for 6 years from 2013 to 2019, among which 4 veterinarians, 2 animal technicians, and 10 companion animal guardians were infected. A total of 2 people died, 1 veterinarian and 1 guardian of companion animals. This slide is a photo of a veterinarian at the veterinary hospital in Gyeonggi-do, South Korea, very nearby the Seoul, presenting his experience in 2019 while treating a dog who had been bitten by ticks and had severe clinical symptoms, bloody feces and hematemesis, thrombocytopenia, and abnormal values of the blood test. The dog patient died within 2 hours. The veterinarian showed severe clinical symptoms after 10 days and was admitted to the intensive care unit of the university hospital and was diagnosed with SFTS virus infection. It is strongly presumed that the veterinarian who treated the patient was infected with a secondary infection through close contact with the infected dog. This research report is a study conducted by the Korean Disease Control and Prevention Agency, and is a serological survey of the SFTS virus in domestic animal hospital workers. From January to May 2021, 103 animal hospital workers' serum samples were tested for SFTS using ELISA, IFA, and neutralizing antibody test. And the results were positive among the participants for 3.9%, 2.9%, and 1.9% tested positive by each test method. Therefore, it is presumed to be a hospital-acquired infection, and it seems that personal protective equipment should be worn thoroughly during treatment. This data is an initial pilot project study in 2022, which investigated the antibodies to the SFTS virus in local small animal clinic in Korea that had companion animals infected with the SFTS virus for a year. This study was conducted on 103 animal clinic workers and 1 out of 42 veterinarians, 2 out of 44 animal case workers. 1 out of 9 administrative staff were serologically positive. This means that it is suspected that there was a secondary infection caused by close contact with companion animals without a person knowing it, and a large-scale study is needed in the future. This slide shows the SFTS incidence rate for 100,000 people by region in 2021. And the asterisk is a map showing the locations of participating veterinary hospital. Also, these data are preliminary and limited in scope. It is important to note that veterinary workers were serologically positive for SFTS virus. This means that secondary infection occurred through close contact with companion animals infected with the SFTS virus without the patient's knowledge. I'm confident that if a large-scale survey is conducted with the active cooperation of veterinarians, specific data can be presented. This table shows the results of the survey. The key takeaway from this table is that serologically positive people were not wearing gloves, were wearing masks, but not goggles, indicating insufficient PPE. This slide is a case of secondary SFTS infection from animals to humans in China. Between June and November 2020 and 2023, more than 1,500 mink farms in Yantai City, Hyanggup City, and Haiyang City in Shandong Province, China, were investigated. The incidence was 30 to 41%, and the mortality rate of infected animals was 100%. Occurring in all age groups, and deaths occurred 1 to 3 days after loss of appetite. Two people who worked at this mink farm tested positive for SFTS antibodies. The workers at this farm were those who euthanized and skinned minks for pure. This slide shows a case of SFTS virus outbreaks occurring simultaneously in multiple fox farms in China in 2023. Between July and September 2023, SFTS occurred continuously for 2 to 3 months in 6 Arctic fox farms in Shandong and Liaoning Province, China. The symptoms included anorexia and weight loss, and dozens to hundreds of foxes per farm died. Autopsy results showed splenomegaly and bleeding in intestinal organs. Swab samples were collected from the tissues and organs of 13 dead foxes, and genetic tests confirmed the presence of the SFTS virus in all of the samples. In addition, the SFTS virus was confirmed in all environmental samples, including feed containers, serum, and anal swabs also. The SFTS virus that originated from this fox farm was identified as genotype C2. This case report was published in the New England Journal of Medicine in 2012. Two men from northwestern Missouri independently presented to a medical facility with fever, fatigue, diarrhea, thrombocytopenia, and leukopenia, and both had been bitten by ticks 5 to 7 days before the onset of illness. Ehrlichia sharpinis was suspected as the causal agent, but was not found on serological analysis, PCR assay, or cell culture. They tried to culture isolation of the virus. Electron microscope revealed the virus constantly with members of the Borna virus, Borna viridae family. NGS and phylogenetic analysis identified the virus as novel members of the flavivirus genus. Both viruses isolated from patients were very close to the SFTS virus. These findings suggest that tick-borne SFTS virus may be circulating in the natural environment of the United States, and surveillance should be conducted. This slide is a summarized schematic diagram based on the study of the transmission cycle of the SFTS virus in nature. The ticks are competent vectors for transmitting the SFTS virus in transovarial and transstadial transmission mode. Primary infection of the SFTS virus is transmitted to humans and animals by ticks. The secondary infection have been confirmed in human-to-human transmission of the SFTS virus, and also there is evidence of secondary infections between experimental animals species transmitted to the same species of the experimental ferret and beagle dog model. Also, recently veterinarians and guardians were infected with the SFTS virus from hospitalized cats and dogs. These are secondary infection cases from animals to humans in Japan. In China, cases of secondary human infection have been reported from farmed wild animals such as mink and fox. It is a very important public health issue. The government should establish a guideline for the infection of the animal SFTS virus to ensure that there are no secondary infections of animal workers or guardians. There are currently no commercially available treatments or preventive vaccines, and research and development are ongoing. Since SFTS has a very high mortality rate in humans, the government and related companies are investing in basic research and clinical trials to develop vaccines and therapeutics treatments. Human and animal medical staff should wear personal protective equipment to prevent secondary infection when treating patients with suspected SFTS. Ticks and tick-borne disease have never been completely eradicated since they were discovered. So how should we manage ticks and tick-borne disease? Also, many studies have been conducted for a long time to control these problems. A complete tick control program has not yet been developed. Also, many studies have been conducted over a long time to control this problem. A complete tick management program has not been developed. So therefore, government agencies and researchers in the field of humans, animals, and the environment should work together to solve this problem based on the One Health concept. Rapid diagnostic method, therapeutics, and vaccines are needed to protect humans and animals from various tick-borne pathogens, including the SFTS virus. In addition, a program for eradicating ticks, the disease vectors, should be developed. I have received funding from these government agencies to study SFTS virus in ticks and animals. Thank you for your attention.